Bacterial vaccines are relatively straightforward to produce because bacteria possess their own replication machinery. If you place a small seed culture into a simple nutrient broth within an industrial fermenter, those cells will divide exponentially on their own. Viruses lack that internal machinery. They are obligate intracellular pathogens. While surrounding bacterial cells rapidly multiply, a single viral particle floating in that same nutrient broth remains completely motionless. This creates an immediate hurdle for pharmaceutical engineering. You cannot cultivate a viral mass using standard biological fermenters. To grow a virus at scale, you have to provide it with a living host system. Navigating this requirement dictates the entire life cycle of a viral vaccine, from initial cultivation in live cells, through chemical attenuation to neutralize the pathogen, to the stabilization of the final dose. The entire pipeline is designed to present a safe viral antigen to the human immune system, with the specific goal of activating CD8-positive cytotoxic T cells. Because a virus is inherently tied to a host cell for its survival, mastering this living host manufacturing pipeline is the only physical way to engineer a safe, long-term immune response. To achieve mass viral cultivation, manufacturers rely on two primary biological ingredients: continuous cell culture systems and embryonated eggs. The viral seed strain is injected directly into these living tissues to initiate the replication cycle. Once inside, the virus commandeers the host's cellular machinery. It hijacks the native ribosomes and enzymes, forcing the host cell to halt its normal functions and rapidly assemble thousands of new viral genomes and protein structures. As the newly assembled virions push against the cell membrane, the host cell stretches and eventually ruptures, releasing a massive wave of new viral particles into the surrounding biological fluid. This creates a significant engineering problem. The resulting culture fluid is now rich with the target virus, but it is also heavily contaminated with ruptured host cell proteins, lipids, and biological debris. While living host systems solve the problem of mass cultivation, they mandate an aggressive, highly precise downstream purification process to ensure the final product is safe for human injection. At this stage, the raw, newly cultivated virus is highly pathogenic. Before it can be utilized, it must be structurally modified into one of three distinct vaccine architectures. The first is the live attenuated vaccine. Through serial subculturing or targeted genetic modification, the pathogen is weakened into a dormant state, drastically reducing its virulence while keeping the organism alive. The second is the inactivated vaccine. This approach uses direct chemical intervention to completely neutralize the virus's ability to infect cells. Vaccine manufacturers introduce harsh chemical agents, specifically formalin or beta-propiolactone. These molecules chemically cross-link with the viral RNA or DNA, destroying the pathogen's genetic core. While the internal genetic core is shattered into useless fragments, preventing replication, the outer antigenic surface proteins remain structurally intact, providing the markers the immune system must recognize. The third architecture relies on subunit or recombinant design. Subunit manufacturing bypasses the whole pathogen entirely. Instead of modifying the entire virus, engineers extract and utilize only the isolated antigenic proteins, leaving the infectious components behind. The chosen attenuation method, whether genetic, chemical, or recombinant, directly dictates every subsequent step of the vaccine's formulation and its strict environmental storage limits. Now that the virus is structurally safe, it must be separated from the host cell debris generated during cultivation. Engineers run the biological fluid through extensive physical processes, specifically ultrafiltration and chromatography, to isolate the target antigen and ensure absolute sterility. The purified antigen then enters the formulation phase, where specific pharmaceutical compounds are integrated into the solution to create the final dose. These additives serve two roles. Stabilizers protect the fragile viral envelopes from degradation, while adjuvants, such as aluminum salts, are added to intentionally amplify the local immune response upon injection. At this stage, the liquid has transitioned into a stabilized, precision-balanced pharmaceutical product ready for clinical use. Once injected into human tissue, the vaccine encounters the immune system's initial scouts. The engineered viral particles are physically engulfed by antigen-presenting cells, or APCs. The APC processes the viral antigen and presents those markers to the broader immune system, triggering a critical divergence in the cellular response. The first pathway activates B cells. These mature into plasma cells, which begin producing vast quantities of specific neutralizing antibodies, particularly immunoglobulin G, or IgG. The second pathway is vital for handling viral threats, the activation of CD8-positive cytotoxic T cells. Because live viruses survive by hiding inside host cells, antibodies alone cannot clear an infection. CD8-positive cells are required to hunt and destroy the infected host cells themselves. Achieving this simultaneous dual activation, B cells for antibodies and CD8-positive cells for cellular destruction, is the biophysical goal of the vaccine, resulting in the formation of long-term memory cells. Despite this sophisticated biological engineering, all viral vaccines share a severe physical vulnerability. Their complex proteins, lipid envelopes, and mRNA sequences are thermodynamically fragile. For most inactivated viral vaccines, stability is maintained within a strict storage window of +2 to +8 degrees Celsius. If temperatures exceed 8 degrees, molecular motion increases. The engineered protein antigens begin to vibrate and unfold, entirely losing the specific shape that the immune system needs to recognize. A drop below 0 degrees is equally destructive. Freezing temperatures cause jagged ice crystals to form, which puncture the viral structure and force the aluminum adjuvants to clump tightly together into irreversible aggregates. Other architectures require different thermodynamic constraints. Live attenuated formulas often require freezer storage at -15 degrees Celsius to keep the organisms dormant, while mRNA formulas may require -70 degrees to halt enzymatic degradation. To combat these thermal threats, the pharmaceutical industry relies on the cold chain. This logistics network utilizes heavily insulated equipment and vaccine vial monitors, labels that permanently change color upon critical heat exposure. Once protein denaturation or adjuvant aggregation occurs due to improper storage, the vaccine is permanently compromised. That loss of potency cannot be reversed. A successful immune response is a logistical victory as much as a biological one, requiring an unbroken chain of precision engineering from the initial bioreactor to the final temperature-controlled vial.